Well, today I'm going to be talking about two independent projects, but I'm going to try to combine them or connect them in this talk. First, I want to start by talking about habitat loss, because I found that surprising that habitat loss is the most important threat for biodiversity nowadays. And that's the same throughout geological time. So there is the sea level changes associated with habitat loss have been widely associated with extinction events through time. After the last mass extinction event and throughout the Cenozoic, the most dramatic changes in sea level occurred in the Pliocene and Pleistocene epochs. And even though these changes in sea level have been associated with some regional extinctions, for example, of some corals in the Caribbean, it has been widely stated that vertebrates remain resilient or resistant to these environmental changes. However, we know that there have been some examples of isolated extinctions during this time. For example, the extinction of the biggest shark that ever lived, that I'd worked in most of, Megalodon, became extinct around that time. Also some species of sea turtles, seabirds, and marine mammals. There was a significant decline in the diversity of cetaceans around that time. So our research asked the question whether these extinctions were isolated examples of independent extinctions, or if they were part of an extinction event. So to answer this question, we assessed extinction rates through time and assessed if there were higher extinction rates around this period. The next question was whether these sea level changes cause these extinctions, or most specifically, if these changes in sea level cause habitat loss. And finally, our last question was about the effects or the ecological effects of these extinctions. Particularly, we wanted to know if there were any changes in functional diversity around this time. And this is where I'm going to connect the other project, because we're comparing now the effects of this extinction of marine megafauna, big animals, with those of small invertebrates in the Caribbean. So to answer the first question, we gathered a big data set from the entire Cenozoic in the paleobiology database, and we gathered all the occurrences, as opposed to first and last occurrence of each taxa. And we used a software called PyRate, Pi for Python and Rate for Rates, that Daniele Silvestro, one of our co-authors, developed. And with this software, we use all the occurrences, and we calculate times of origination and extinction at the same time as preservation probabilities and sampling probabilities, and with that, extinction rates. We found that during the Pliocene, there were higher extinction rates than during the rest of the Cenozoic. In fact, the Pliocene extinction rates were 3 times higher than the mean Cenozoic level. We did a better resolution kind of analysis, and we found that specifically at the end of the Pliocene is where these extinctions were higher, very close to the Pliocene-Pleistocene boundary. When we look at the proportional extinction, we found that each group of megafauna, I have to say that megafauna includes marine mammals, seabirds, marine turtles, sharks, and rays. When we combine them all, there was a proportional extinction of 36%. So around 1/3 of the taxa became extinct around this time. Now the next question was about changes in sea level and whether or not they resulted in habitat loss. So what we did is to use these sea level curves, and for each of these sea level scenarios, I think the time scale was 100,000 million years, we calculated the area available, the coastal area. So we calculated the bathymetry, and we selected the areas that were up to 200 meters, and we regarded that as coastal habitats, and we quantified that area. And this is what we found. So the Pliocene available area, the mean area, was much higher than the one in the Pleistocene. But when we look at the specific changes per each sea level scenario, we found that there was a dramatic drop in area at the end of the Pliocene. And after that, there were very violent fluctuations. We hypothesized, or we proposed, that these changes in sea level and in area or in habitat cause these extinctions. And this hypothesis was reinforced by some trait-based analysis we did. I'm going to explain this trait-based analysis later in the next slide, but for now, just know that we assigned these traits to each genera. And we performed some generalized linear models, and we found that thermoregulation was the trait that better predicted this extinction, with animals that were mesotherms and endotherms, so those that are able to control their body temperatures, at least at some degree, having higher chances of going extinct than poikilotherms. The reason why we think this reinforces our hypothesis is because endotherms and mesotherms, because they are homeothermic animals, they have higher metabolic demands than poikilotherms. And because of that, they need more food. And because of that, they need more area. Therefore, a loss of area is going to affect them more, as opposed to poikilotherms, for instance, they will be more affected by temperature per se. Now, in terms of the effects on functional diversity, we use an approach that's been used in ecology. We use the methods of Mulliot in 2013, in which you have a species pool. So we use the species pool we gather from the paleobiology database, and we assign these traits. So the categorical traits that we assigned were maximum body size, gill, that we call gill, or the type of diet, macropredator, herbivore, micropredator, and so on, habitat, zone, and thermoregulation. So we assigned these traits to each of these taxa, and we created a functional space based on a principal coordinate analysis. We also did some presences and absences, because in this approach, you create one single space, and then in that single space, you quantify, based on the presences and absences, the volume of two different communities, and you compare those volumes. In this context, a shift will be the lack of overlap between two functional spaces. We did that, and this is what we found. So this is the functional space for the Pliocene before the extinction and the Pleistocene after the extinction. Each dot here represents not a species or a genus, but a functional entity. A functional entity is a unique trait combination. For example, this is a functional entity that is right here, and it has a body size of more than 12 meters, is a macropredator, is in the pelagic zone, coastal habitat, and mesotherm. This particular functional entity is filled by one genera, and one species actually, which is Megalodon. We found that after this extinction event, we lost 7 functional entities, so 7 of these dots didn't make it to the Pleistocene, and also 4 evolved. Now, these plots here that don't take into consideration sample differences, but in fact, the sample size of the Pliocene and the Pleistocene were different. To cope with this, we did some bootstrapping, and we standardized the sample size of both, and we run a bunch of iterations. And we found that once you take that into account, there was a loss of 15% of functional richness or functional space, and also 20% of shift. So to summarize, we have that we lost 1/3 of of taxonomic diversity. We found that we lost 15% of functional richness and a 20% of shift. So these numbers may seem modest or not a big of a deal. However, when you compare this with other studies that have done similar approaches to assess ecological consequences of extinctions, we find that actually they're very high. For example, this paper by a colleague in the Naturkunde Museum found that after the mass extinction event of mollusks, you only lost 1 functional entity. So the reason why we think we have such a difference is because we're talking about marine megafauna. So these are big animals. And actually, because they're so big, they're not very redundant. So for each functional entity, we have, for the most part, 1 or 2 taxa filling that space or filling that particular group. So of course, when you lose one species, you lose functional entities. So we wonder if that's the case for mollusks. We couldn't do some sort of direct comparison with other studies because it depends on what traits do you use to do that, what resolution, etc. So in case of mollusks, this is the kind of the theory we're working on. You have a range of functions just with marine megafauna, but because they're smaller, they tend to be more redundant. So for example, if we take that these boxes here are functional entities, we'll see that for mollusks, we think, or the hypothesis, that we have a lot of species in each of these boxes. When you remove some species, you don't lose these functions because there's still at least 1 species or genera playing that function. But if you remove other 10 species in this community, then you do lose some functional entities. So we wanted to know if this was the case or this difference was evidence in this other extinction event. So this other extinction event I'm going to talk about is a well-known extinction event that occurred in the Caribbean. It happened around the Pliocene and Pleistocene as well. And it's been said that 70% of benthic community of benthic species went extinct, or genera, sorry. And why? Because when the Isthmus of Panama formed, the oceanographic conditions of the Caribbean changed, and this caused this extinction. This is debatable now. So we gathered a huge data set of Caribbean mollusks, and we run the pyrite analysis. Indeed, this extinction took place in the Pliocene, Pleistocene with our methods, and we found that for mollusks, there's an extinction of 35%, only for mollusks. So it's very similar to what we found with the marine megafauna. Interestingly, or in agreement with our hypothesis, we found that changes in functional diversity were almost not seen. I mean, we were almost negligible. So when we see the changes in functional entities, it doesn't change much during the last 23 million years, and not during the extinction event. Same for redundancy, and same for functional richness, which is the volume of the space. So let's ignore these lines for now, and just this one is the volume. So as a home take message, what we found is that there was 2 extinction events. One was global, one was regional. In both, we lost a similar amount of genera. However, with the marine megafauna, we lost around 7 functional entities, and for mollusks, only 1 or 2. And we think that is because redundancy is a very important determinant. So for example, the redundancy of marine megafauna is 1 or 2 genera per functional entity, whereas for mollusks, we have at least 4. So we propose that functional redundancy is a key element in assessing the ecological impacts of extinctions. And with that, I would like to thank my collaborators, and actually for the modus part, I'm sharing first co-authorship with Christine Bacon. And thank you to my postdoc hosts. Thank you.